Pharmacodynamics is the study of effect of drugs on the body. So in this basically we study what are the effects of the drug and how does the drug carry out those effects. Okay, so when we talk about the meaning of the term drug effect, are we actually referring to? Drug effect basically is changing the physiological process. It can be changing the rate of any physiological process or the magnitude, how much magnitude of any physiological process. So basically, drug itself hardly has its own very different action. In fact, whatever physiological processes are there, it either increases or decreases the rate and magnitude of these physiological process, and based on this, whether it is increasing or decreasing the effect, we have drugs which act as agonists and antagonists. We will talk about these a little bit later. Okay, now how do the drugs act? Well, mechanism of action of drugs mainly is by three mechanisms. One is either it can act without binding to any receptor, that is by virtue of simply its physical or chemical property. Okay, so it doesn't bind to any receptor in the body, but by its physical and chemical property it has its action. For example, activated charcoal. Activated charcoal has adsorptive property, and that is why it can be used to remove poisons. Then there are laxative, the bulk laxatives. They act by just their physical action. There is other action as well, but the physical action provides the mass. Okay, then certain antacids which neutralize the gastric HCl, the hydrochloric acid which is released in the gastric juice. So for that neutralization, the antacids maybe mainly because of their basic nature, they neutralize it. Okay, then chelating agents are there. Chelating agents remove heavy metals like calcium by binding to them. Okay, then second mechanism which majorly we will focus on is by binding on receptor or some other proteins on host cell. Okay, and the third mechanism is by binding to receptor on microbes. Okay, so might be some proteins are there on the microbes, and mainly the antibiotics which act on microbes, so they act by binding to the receptor on the microbes. Let us focus on the receptor on host. So the effect of this depends on the receptor specificity. That means which receptors in the body the drug is binding to. Okay, is it binding to only one type of receptor or it is binding to many receptors? So when it binds to only a particular receptor, only one type of receptor, that means it is more specific. Okay, then what is the tissue specific expression of the receptor? So suppose a particular drug binds to a particular receptor. If that receptor is present in many tissues and maybe the action of the drug is for a particular tissue only, then okay, it will have the action, but it will also have the side effects because that particular receptor is present on other drugs. Okay, so that is tissue specific expression of receptor. And obviously if you want that certain drug should act on a particular tissue, then the protein on which the drug should bind should be present on that particular cells of that particular tissue. Okay, then drug access to target tissue, that means the drug should reach that particular target tissue. For example, if a drug has to act on neurons, then drug should reach the neurons. It should cross the blood-brain barrier. Then what is the different drug concentration in different tissues? So even if the drug is reaching to the target tissue, what is the concentration in various tissues? So depending on that, the effect will be there. Then it can interact with other drugs, and the effect of the drug can also vary from person to person based on genetics, that is the pharmacogenetics. In detail we will talk about pharmacogenetics in some other video. Now, little bit about the specificity and drug response. So, I am telling here that the drug binds to particular protein, and this binding of the drug to a particular protein, the strength of binding is known as affinity. Okay, so which particular protein it is binding, that is known as specificity, but the strength of binding is measured by affinity, and there is a measure of that known as dissociation constant. So suppose if the drug is binding weakly, then the probability of dissociating from this protein will be much more. So if we see in a particular time, it will remain in dissociated state, that means separated from the protein for much more time than in the associated state, that is in bound form. Okay, so this concept helps us in measuring the dissociation constant, which is a measure of affinity. More the affinity, binding time will be more, the probability of dissociation will be less. Okay, and this binding basically is also dependent on the chemical structure of the drug. So this chemical structure, I told you, determines the affinity. Then it also determines the intrinsic activity of the drug, that is known as the efficacy. That means, okay, it is going to bind to the drug, but is there any activity of the drug? Is it going to change the function, or is it just binding the protein? Is it going to alter this protein structure now so that the further actions of the drug can take place? Okay. And third, I told you, is the specificity. That means which receptors it is binding. Is it binding only a particular type of receptor? And is this receptor spread throughout the body? Or there are multiple receptors which the drug is binding. Say suppose for a particular ligand in the body, we have different types of receptors. Say adrenergic receptor can be alpha 1, alpha 2, beta 1, beta 2, beta 3. So if we are giving certain drug which is mimicking the action of sympathetic system, is it binding to only alpha receptors or only particular type of alpha receptor, or it is binding to all types of receptors? So depending on that, which receptor it is binding, the drug action can occur. And in fact, by changing the chemical structure of the drug, we can change that which receptors it can bind to. So when we are studying about individual systems in pharmacology, then we will see that we have different types of drugs which are either very specific or they are non-specific. So that depends on the chemical structure. Also, the other properties of the drug can depend on this chemical structure. For example, how much it crosses the cell membrane or how much it is metabolized. All can be little bit altered by changing the chemical structure of the drug. Now, where can the drug bind? So, I have told you that there are receptors or there are proteins. Now, these receptors or proteins can be present outside on the cell membrane, or they can be present inside the cell as well. And the drug binding to this, it can be on a site on this protein where maybe a natural physiological ligand is present. So that is the main site. Okay, it can bind there also, or it can bind to a nearby site also where the natural ligand is not binding, but there is a nearby site, and this is known as allosteric site. So here the drug can bind. Okay, or it can cross the cell membrane and it can bind to a protein within the cell, or it can also bind with the plasma proteins, and binding with the plasma protein affects the pharmacokinetics of the drug. So this part I have already dealt in pharmacokinetics. You can watch those videos. So yes, so drug can bind at various sites, and we will see that where are the drug binding sites. So where are the major proteins where the drug can bind. Before that, I just want to differentiate between certain terms known as drug action versus drug effect. Well, drug action is basically binding of the drug to the protein. So that is the drug action. Only the concept of binding of the drug with the protein. Okay. On the other hand, drug effect is that once it binds with that protein, what is the further downstream events taking place, which is ultimately leading to the physiological response. Okay. So this is what is happening to the receptor, whether any conformation change in the receptor is happening or it is simply blocked. Okay, so we can say that this is basically affinity, telling about the affinity, and this is telling about the intrinsic activity of the drug. Okay, so these terms we have already seen before, affinity and intrinsic activity, or also known as efficacy, right? Now moving on to that, what are the various proteins to which the drug can bind? So there are fundamentally 4 different types of proteins to which the drug can bind. There is interaction with the enzyme, there is ion channels, then there is transporters, and there are receptors. Now, apart from these, there are certain proteins to which the drugs can bind that are like very less components, so we are not dealing it separately, but I'll just mention here, for example, certain drugs like taxanes can bind to structural proteins. So I have not put it in this classification. So structural proteins like tubulin, and these are anti-cancer drugs. Okay, then there are certain drugs like alkylating agents, they can bind on nucleic acid as well. Okay, so apart from this also, the drugs can bind to certain other proteins as well, even nucleic acid. So we will majorly focus on all these four types of receptor drug interaction. So one is enzyme interaction. So there can be an enzyme to which the drug can bind. For example, statins. Statins can bind with HMG-CoA reductase. Okay. Then there are ACE inhibitors. ACE inhibitors can bind with the your ACE enzyme. Okay. So these are the enzymes. Now, after binding, either they can be enzyme stimulation, or they can be enzyme induction, or they can be enzyme inhibition. Now, enzyme stimulation means that whatever enzyme is present, now that particular enzyme starts acting more. Okay, so this type of enzyme stimulation, that means increased activity of the enzyme which is present in the body, this is rare. The drug action by this mechanism is rare, because normally only the enzyme generally operates at its maximum capacity in the body. Okay, so this is rare. However, there is another type of enzyme stimulation, that is enzyme induction. Enzyme induction is where there is synthesis of more enzyme. Synthesis of more enzyme. One example of this is like barbiturates. Barbiturates induce the synthesis of enzyme UDP glucuronosyl transferase. Okay, so this is present in liver, and that is for glucuronidation of the various substances. So UDP glucuronosyl transferase by barbiturates. Then we have enzyme inhibition, where the activity of the enzyme is decreased. So when this is happening, how does the body respond? To understand that, we will just visit briefly the concepts of Km and Vmax. What is this? So when there is an enzyme, say suppose this is an enzyme, when this enzyme reacts with a substrate, okay, now because of this reaction, there is production of enzyme. Enzyme gets separated, and that substrate is changed into the products. Okay, so initially we have enzyme which is free, and whatever its substrate is there, it can bind with that and it can form the enzyme substrate complex. And because of the formation of this enzyme substrate complex, some reaction will occur where the substrate will be converted to the product and enzyme will be released. Now, there is the rate of this reaction, right? And the maximum rate which can be achieved, that is known as Vmax. That is the maximum velocity of the reaction. After this, the velocity is not going to increase. So what we see, say suppose on x-axis there is substrate concentration, and on y-axis we have the rate of reaction. So we see as the substrate concentration is increasing, that means this substrate is increasing, the velocity of reaction is increasing, but ultimately it reaches Vmax. And why this velocity of reaction increases? Because say suppose we have lot of enzyme, right, and the substrate is there. Initially the substrate is less, so only this substrate is going to bind to this particular enzyme. We don't have any more substrate to bind to another enzyme, right? So as the substrate increases, it is going to occupy more and more amount of the enzyme, and hence we get a increase in the rate of the reaction. Ultimately, all the enzyme will be occupied and we have a maximum velocity of the reaction. So that is known as Vmax. Now, the substrate concentration at which the velocity is half of Vmax, that is known as Km. What is Km? Substrate concentration at which the velocity of the reaction is half of Vmax, that is known as Km. So we have one parameter for substrate concentration and we have another parameter for rate of reaction. So we'll talk about two things, Km and Vmax. Now this diagram tells about what happens to Km and Vmax when the drug binds with a particular enzyme. Okay, now that can be enzyme stimulation, enzyme induction, or there can be inhibition. So suppose there is enzyme stimulation, which I said that it is rare. What is going to happen? That the same enzyme, now the rate of reaction is going to increase and the Km also decreases. So we have the Vmax, you see here, this is the normal one, and say suppose this is Vmax for this particular condition and this is the Km. Now, when there is enzyme stimulation, you see how the graph is changing. That means the Vmax is increasing, the rate of reaction has increased that much, and also the Km has decreased. That means the substrate concentration to cause the maximum to this half max where it can be reached, that has also decreased. Okay, less substrate is now causing the reaction to be faster, right? Then we have enzyme induction. In enzyme induction, the enzyme quantity has increased, the amount of the enzyme has increased. Now, when the enzyme is induced, what we see that the Km does not change, right? So, you see Km does not change, Km is the same, but the velocity, the Vmax increases. So, because this Vmax is increasing, what is happening? That the half Vmax is also more, right? So, at this particular Km, the half Vmax is more because the Vmax itself has increased. So when the enzyme is induced, we see that the Km is not changed, but Vmax, velocity of reaction, is increased. So same amount of concentration of the substance, but rate of reaction increases, right? So that means suppose there is some substance which needs to be removed from the body, and now this phenobarbital is inducing the enzyme. So that particular amount of the substance is enough to be removed. So Km is not changed. We need not increase the amount of the substance for the enzyme induction to for this reaction to occur. Okay, so Km is not changed, but with the same substrate concentration, the Vmax, the velocity of reaction, has increased. Now coming on to the inhibition. Inhibition can be of two types, that is competitive inhibition and non-competitive inhibition. So here is the enzyme schematic diagram, and we have two sites, the catalytic site and the adjacent site. Now catalytic site is the one where the substrate is going to bind, which I have already told before. Now the drug which you give, if it binds on the catalytic site and doesn't allow the substrate, the original substrate, to bind to the catalytic site, that is known as the competitive inhibition. On the other hand, suppose the substrate easily binds, whatever substrate is there that can bind, but the drug which you have provided binds to this adjacent site, here it binds, then that is known as the non-competitive inhibition, because now the drug is not competing with the substrate to bind on the catalytic site. Okay, so two types, competitive inhibition, non-competitive inhibition. Now in competitive inhibition, suppose if we increase the concentration of the substrate because it is competing with the drug, what will happen? Since you have increased the concentration of the substrate, now this substrate will become more active in binding to the catalytic site, because the more amount of the substrate is available, and say suppose the drug amount is only this much, so when the drug will dissociate, it is not permanent binding to the catalytic site. Remember that there is always association and dissociation. So this is known as reversible binding, right? So that is competitive inhibition. So now if the concentration of the substrate is increased, then as it dissociates, the probability of binding of the substrate is more because we have more amount of the substrate. So in competitive inhibition, what we see that Km increases. That means if you see the amount of the substrate you increase, then the reaction is going to take place, but Km increases but Vmax doesn't change. Okay, so you see this graph of competitive inhibition, what we see here, that Vmax, both are ending at the same point, Vmax is same, but Km has shifted to the right side. So that is Km and half Vmax is same. Okay, so that is competitive inhibition. And non-competitive inhibition, when it is binding to the adjacent site, well, it doesn't allow the reaction to take place. Even the substrate is able to bind, but the reaction is not going to take place. So what happens that Km remain unchanged, but Vmax is decreasing, and because Vmax is decreasing, half Vmax is decreasing, so for this half Vmax the Km doesn't change. Okay, so Km remains the same. That means the substrate concentration at which half maximum velocity is reached. So that is non-competitive inhibition. Before we proceed, I just want to talk about this competitive inhibition, which can be of two types. That is the equilibrium type and non-equilibrium type. Okay. Equilibrium type is the one which we just discussed. That means there is reversible binding of the drug on the catalytic site. So it is going to be replaced also, and because of this, the Km is going to increase. More substrate concentration will be required for the same Vmax to be achieved. But if the substrate concentration is more, then the reaction can proceed. On the other hand, non-equilibrium type, what happens that there is a very strong bond between the drug. Here the drug will bind very strongly with the catalytic site, and that is irreversible binding. Okay, irreversible binding of the drug to the catalytic site. So that means now even the substrate concentration is changed, it is not able to bind to that catalytic site. Okay, so in this case Km is increased and Vmax, I'm writing other side, so non-equilibrium type, Km is increased and Vmax also is decreased, because now the substrate is not able to bind. The enzyme will never achieve that Vmax, that is the maximum rate of the reaction. So that is non-equilibrium type. Example is organophosphate drugs. Organophosphate drugs bind with this esteritic site of cholinesterase enzyme. Okay, so in particular different chapters we are going to see about these various drugs which act by various mechanisms, right? So that was about how the drug acts on enzyme. They can be enzyme stimulation, enzyme induction, they can be competitive inhibition, which can be of equilibrium type and non-equilibrium type, and they can be non-competitive inhibition as well. Okay, moving on to the next type of mechanism of action, that is binding to ion channels. In physiology, we know that ion channels are of various types, that is ligand gated, voltage gated, stretch sensitive ion channels. Okay, so ligand gated means, so there is a channel, say suppose this is the membrane, and this channel will have a binding site for a particular ligand, and only when this ligand acts on the channel, it binds on the channel, then this channel is going to open. So that is ligand gated channel. Voltage gated channel are that there is a channel on the membrane, and when there is voltage change, say suppose there is depolarization, then there is opening of this channel. Okay, then we have the stretch sensitive channels. That means if there is any stretch on the membrane, say suppose there is some stretch here, this will cause the change in the shape of the channel and the channel is going to open. So these are the three major types of channels, and various drugs can act on these channels. For example, ligand gated channel, that is present at neuromuscular junction where acetylcholine binds. So there can be certain blockers which act on this channel, neuromuscular blockers. Then we have the voltage gated channel. Say suppose there is voltage gated sodium channel. So we have local anesthetics which act on these channels. Then we have stretch sensitive channels. And the last one which I am referring here because KDT has given, that is G protein regulated channels. Actually this G protein regulated channels, they will come here when we talk about the receptor. But what happens that because the drug acts on G proteins, and then via the downstream changes, say suppose this is the cell and here there will be the G protein. Okay, now when the drug binds here on the G protein, then there are certain changes within the cell, and then via these changes there is opening or closing of the nearby channel. So these are ion channels are known as G protein regulated channels. Okay, so this is the G protein coupled receptor only. The drug is acting via binding to the receptor, not via binding directly to the ion channel, right? Next is binding to certain transporters on the cell. Okay, so say suppose we have different transporters on the nephrons. Sodium potassium 2 chloride transporter is there, sodium chloride. This is present on ascending limb of loop of Henle. Sodium chloride, that is presented on DCT, and there might be drugs which bind to these. For example, thiazide diuretics bind to this. Okay, then loop diuretics bind to this and they block these channels. Then there are uptake transporters. For example, say this is the neuron and it releases the neurotransmitters. But these neurotransmitters are taken back into the nerve terminal as well. Now, for this taking back of the neurotransmitter into the nerve terminal, we have various transporters. So those can be blocked. For example, we have SSRIs. So they can block the reuptake of the serotonin. Then block reuptake of GABA. For example, certain anticonvulsants are there which can block the reuptake of GABA. Okay. So these are the various mechanisms which we will be dealing in various chapters. So this is basically the fundamental concept. You should know that how the drugs are acting. Then moving on to the receptors. In receptors, we have various types. For example, ion channel linked receptor, G protein coupled receptor, and enzyme linked receptor. This ion channel linked receptor is the same what we read before, that is the ligand gated channel. Ligand gated channel. So this is what actually ligand binds to the channel and then the channel opens. So that means the receptor, this is the receptor where the ligand will bind. So this receptor is basically linked to a channel. So that is why ion channel linked receptor, or we can say as ligand gated channel. Okay. Then we have the G protein coupled receptor. G protein coupled receptor in turn can act by activating adenyl cyclase, phospholipase C, or directly by causing the opening or closing of other channels. And then we have the enzyme linked receptor, that is the receptor tyrosine kinase, serine threonine kinase, and the guanyl cyclase. Now, this concept, what are the various types of receptors, I have dealt in another video on mechanism of hormone action. Full video is there for physiology, so that you can see. I will add in this playlist as well. Okay. So those were the various mechanism of action of the receptors. Now, when we started the video, I told about that based on this interaction with the proteins, we have drugs which are known as agonists or antagonists. And in this agonist and antagonist, we have different types of agonist and antagonist. So agonist is a drug which is increasing the action, which is whatever is the physiological action, it is increasing that action. And antagonist is a drug which is blocking that action. So we will talk about this agonist, antagonist, and what are the various theories which explain this agonist and antagonist in the next video. Thanks for watching the video. If you liked it, do press the like button, do share the video with others, and don't forget to subscribe to the channel Physiology Open. Thank you.